welcome back. Don't forget to like, share and subscribe to my channel. We're looking at the digestive system in the organisation topic, and today we're going to look at enzymes and the factors that affect them. Enzymes need the right conditions to work properly. Factors that can affect enzymes are temperature and pH. Both temperature and pH will affect the rate of a reaction. Let's have a look at temperature to start off with. Increasing the temperature will increase the kinetic energy. Increasing the kinetic energy will increase the collision of the enzyme and the substrate. This ultimately will increase the rate of reaction. If the temperature is too hot, that will affect the bonds holding the enzymes and it will break them. This breaking of the bonds will change the shape of the enzyme's active site, in which case the substrate will not fit into the enzyme and we say the enzyme becomes denatured. Another factor that will affect the enzymes is pH. With the different pHs, it interferes with the bonds of the enzymes, the bonds which are holding the enzymes together. If the pH is too high, this will change the shape of the active site on the enzyme, and this ultimately will again make the enzyme denatured. All enzymes have an optimal level that these factors can work on. Optimal level is where the enzyme is working best at. Illustrated in graph A and B, we have a rate of reaction graph for both temperature and pH. The optimum level for temperature is here, and for the pH it is here. This is where the enzyme is most active. We can see that the rate of reaction increases as the temperature increases. At this point it becomes optimum, meaning that it works the best here, and then the rate of reaction drops. So this means at this point it must be denaturing. Same with the pH. We can see our optimum level would be at pH 7. So this means at pH 7 the enzyme works best. Past pH 7 we can see the enzyme is now denaturing. We can have different enzymes working at different pHs. For example, we could have an enzyme that may have optimum pH at pH 2. One example of this is pepsin, and this works in the stomach. This means that the enzyme works in acidic conditions because pH 2 is acidic condition. A tip to remember is that the optimum temperature for most human enzymes is around normal body temperature, which is about 37 degrees. For the pH, the optimum pH can be different. For stomach enzymes, it is low pH. We have enzymes that can work in high pH, such as in the small intestines. This brings us to the last section, exam questions. Question 1, worth 4 marks. Identify factors that may affect the rate at which an enzyme works at, and explain the term optimum. Explain briefly what would happen if the optimum level were exceeded. That's 4 marks, so the factors would have 2 marks there. Explaining optimum would have a mark there, and what would happen if the optimum level were exceeded, a mark there, which makes our 4 marks. Let's think back to the factors. The factors that we looked at were the temperature and the pH. Explain the term optimum. Remember, optimum meant where the enzyme was working best at, so it is the level where the enzyme works best at. And what would happen if the optimum levels were exceeded? So remember our graphs, we had one for the temperature, and we had 1 for the pH, remember? So what would happen is that if the optimum level was exceeded, the enzyme would denature, and denature is where your last mark is. So you have 2 marks there. You have a mark for saying where it works best at there, and you have your mark for saying it denatures for your 4 marks. Question 2. Trypsin is a protease enzyme. Trypsin will digest a protein. In an experiment, students investigate a digestion of a protein with trypsin. The students used 6 different concentrations of trypsin. The rate of reaction was calculated from the time taken for the protein to be digested. The graph shows the students' results. On this side we have the rate of reaction, and at the bottom we have the percentage concentration of the trypsin. The question is, describe the relationship between the concentration of trypsin and the rate of reaction, worth 2 marks. If we have a look at the graph, we can see that as the percentage concentration increases, so does the rate. You can see the rate is going up. So we say it is directly proportional. Our answer, we can say the relationship is directly proportional. This means as the percentage concentration rises, so does the rate of reaction. Next section, we need to use the graph to predict the rate of reaction with 4% trypsin, and this is worth 1 mark. To do this, we would go at 4%, we would follow it up, and the best way to do this is to use a ruler. With a ruler, wherever it hits the line at 4%, you can draw a line across, and we can see this is approximately between 0.3 and 0.4. For our answer, we could write 0.35. The word for today is digest. When we think of digest, we should think of 3 different food molecules: carbohydrates, proteins, and fats. When we think of carbohydrates, we should think of carbohydrases as our enzymes, and one of those enzymes is amylase. When we think of amylase, we should think of starch. When we think of starch, we should think of simple sugars, as that's what they are broken down into. We think of proteins, we should think of the enzymes proteases. One such enzyme is pepsin. We think of proteases, we should think of amino acids. When we think of fats, we should think of enzymes such as the lipases, because fats are also sometimes called lipids. When we think of lipases, we should think of fatty acid and glycerol, because these are the components that they are broken down into. Thank you for watching. Join me on my next video. Don't forget to like, share and subscribe to my channel.